Greetings. We live in challenging, somewhat bumpy times, and I'm going to talk for a few minutes with you about tuning up your immune defense and repair system so that you're in elective protective mode, and why this is essential to survive the 21st century in a healthy way. Now, I am Dr. Russell Jaffe. I was trained in conventional internal medicine and then laboratory medicine at the National Institutes of Health. I have a biochemistry background and PhD, and for the last 30 years, I have been investigating how to evoke human healing responses using nature, nurture, and wholeness, using physiology before pharmacology, and by addressing the causes rather than the symptoms of ill health. And when I say the causes, what I fundamentally mean is the lack of the essentials or the excess of the bad. So we want more of the good, less of the bad. We want more of the nutrients, less of the anti-nutrients. We want more of the essentials, less of the forever toxic molecules. And along the way, for disclosure, I am the founder and formulator at PERC Integrative Health. I developed the first ex vivo one-step lymphocyte response assay, the LRA by ELISA Act, which we introduced in 1983. The supplements that are full disclosure and all active were initiated in 1987. And we do have a promising hypertension candidate in trial at RMJHRX. So we're going to look at the fundamentals of immunology, the immune defense and repair system. We're going to look at food and chemical sensitivities and the important relationship between the microbiome, the metabolome, between the central nervous system and the gut nervous system, between the process of metabolism and the process of detoxification. We're going to look at common testing methods, the strengths and limitations of them. We're going to look at the evidence for detecting T cell. These are T-class white blood cells. These are lymphocyte cells that, when they're programmed, react without antibody. They act directly. And there are B cells, known as BRSA, or B-class lymphocytes, and these do produce antibodies. The recent ones are called IgM. The memory ones are called IgG. The mucosal ones are called IgA, or secretory IgA. And there actually is an IgD, an antibody that exists only on the surface of certain specialized cells. So we're going to look at why you would want to have an ex vivo lymphocyte response assay that was reproducible. And we have not just a lymphocyte response assay, we have an HSLRA, high sensitivity lymphocyte response assay, because as you'll learn in just a few minutes, we have better precision on doing a cell culture for delayed allergies than most commercial labs have for measuring things like blood sugar and blood cholesterol. So highly precise, highly reproducible, been subjected to multiple outcome clinical studies that have been successful, and ready for your review. We'll look at inflammation, which is really repair deficit. And much of what we're known for is rethinking the fundamentals of an issue. As a pathologist, as a clinician, I was taught that inflammation is a fire to be fought. And I can tell you, as a physiologist, as an integrative, holistic, functional medicine researcher and clinician, it's really repair deficit. And the repair deficit happens when you lack the essential nutrients, cofactors, and like, and have too much of the forever molecules, the hormone disrupting solvent residues, the toxic metals and mold products, the radioisotopes that all are pro-oxidative. They deplete antioxidants, and we must take in more antioxidants today than in previous centuries because of this burden of antitoxin pro-oxidative material. And what it comes down to from our point of view, and from a clinical individual point of view, is looking at what people eat and drink, what they think and do. And from that, with the help of our smart system and our interpretation of the lab data, we end up with a personalized lifestyle repair program. We use predictive biomarkers and self-assessments. We use proactive primary prevention in everyday practice. So we're upstream, preventing people from falling into the river of disease. We are not focusing on pulling them out by symptom reactive therapies. Now, if we look at autoimmunity, we find it is increasingly common. 23 to 50 million Americans are so diagnosed, actually probably more today. 80% women, although there is a diagnostic bias. There are at least 100 autoimmune-related diseases, and ARDA, the Autoimmune Related Diseases Association, is the group that advocates for more resources and attention to self-attacking autoimmune conditions. But what you really want to focus on is the bottom of the slide, tolerance, which means we're healthy and resilient and able to restore ourselves. When tolerance is lost, self-attack begins. And what we have is the plan, validated and evidence-based, to restore tolerance in the immune defense and repair system, to restore balance in the neurohormonal system, to restore digestive competence in the digestive tract, and metabolic competence throughout the body, so that there can be a communion between the gut nervous system and the central nervous system, between the microbiome and the metabolome, between every part of you in communion with the rest of you. And just to emphasize how important autoimmunity is and inflammation, we see that inflammation and repair deficit is related to arthritis and diabetes and cardiovascular diseases, especially coronary artery disease, vascular diseases, and stroke risk. Cancer risk is now known to be related to immune defense and repair issues, as are pulmonary diseases, especially in the time of mucosal corona type viruses. And then, of course, there's Alzheimer's senility, which is mostly a lack of essential nutrients and an excess of pro-oxidative toxins. Alzheimer's can be seen as diabetes of the brain, and we do use hemoglobin A1C as our marker of carbohydrate metabolism, and we want it to be low. And we'll talk later about the other predictive biomarkers and their role and how to apply them in your practice today, tomorrow, and hopefully forever. And yes, on the bottom of most of the slides, you'll see references either to articles we have written and contributed, or to important people who I would like you to be aware of, such as Scott Grundy, who's the first of the three references here. The third one is one that Jay Srimani and I wrote about rethinking health and inflammation is really repair deficit, in case you want to dig into more detail on that particular topic. Now, immune defense and repair systems have two components, defense and repair. And the easy way to think of it is that during the day, your body is on defense, and when you're getting restorative sleep at night is when it repairs. So being healthy means that we're able to defend ourselves, neutralize any foreign invasion, and still have enough capacity, energetic capacity, metabolic capacity, to repair when we get restorative sleep. So it relates to tolerance in the immune defense and repair system, resilience in the neurohormonal system, balance in regard to what we take in and give out. And we will talk about how to make initial assessments, self-assessment, followed by predictive biomarkers and other detailed tests that will map the territory and identify for you what the needs are. Now, in regard to the immune system, there are two fundamental parts. The innate immune system you're born with. This includes the mucosal elective protective mechanisms at your epithelial barrier, at your surface, at your mucosal surface. It includes your wiggly dendritic phagocytic cells. It includes certain plasma proteins that tune up or call attention to immune defense and repair. And it includes natural killer cells that are responsible for helping identify the abnormal cells that people make and marking them for elimination in a process called apoptosis. And then we have a reserve part of our immune defense and repair system called the adaptive part. And it only operates when the surface and the innate immune system are overworking and needing additional help. And that's where the adaptive cells are called, the B-class lymphocytes, the T-class lymphocytes. And there are many subsets of these that have been identified. We talk about null cells, the CD3. We talk about amplifier helper cells, the CD4. We talk about modulator or suppressor cells, CD8, and regulatory cells. And after a B cell has been programmed to produce a certain kind of antibody, it might become a plasma cell, which is primarily a factory for producing specific antibodies. And so the antibodies from the B cell plasma cells and the T cells combine to be your adaptive immune defense and repair system. Now, the word microbiome is in vogue, and I often use it, and I recommend you do too. We used to just talk about digestion, but now we talk about the microbiome of the digestive tract. And now it turns out there's a microbiome everywhere you look. There's a microbiome in your mouth, and it turns out the best mouthwash is actually ascorbate, native vitamin C, buffered and reduced. And you should stop any alcohol-based mouthwash because it impairs the oral microbiome and increases the risk of central nervous system degeneration. So there's a reason to use ascorbate as your mouthwash. And so whether you look at the skin or the eyes or the nose or the ears or the oropharynx or under the arms, everywhere you look, there is a specialized microbiome that is an ecology of bacteria that are eating things that are being provided mostly by the body, by the excretions in the body, by the secretions in the body, by the access that the body gives it. So there are many microbiomes that are involved with defense and repair, as well as anti-cancer work. And we are leaders in showing you how to restore immune defense and repair competence in every one of those microbiomes. Because it turns out when you do more of the good stuff and less of the bad stuff, every part of the body benefits. And if you're consistent and use the self-assessments and predictive biomarker tests and other evidence that we use to guide practice, the results get even better and better. And one of the results is that people tend to bring, they tend to bring their loved ones and family members, because today, if you're taking symptom reactive medicines, you're shifting problems from one part of the body to another. And what we want to do is get to the cause, get enough of the good stuff in, enough of the bad stuff blocked, prevent the bad stuff from coming in. And we can look at the adaptive immune system in a slightly different way. The natural way is passive maternal protection. The first year of life for an infant is usually protected based on the mother's immune competence. And then active, natural, adaptive immune response to, say, childhood or other infections, where the goal is to have a memory immune response, T cell or B cell response, that will keep you protected if you get exposed again. And then there's the artificial way of activating the adaptive immune system, either with antibody transfer or with active immunization. Think of vaccines and think of people giving antibodies from recovered people to those at risk with the hope of reducing, of giving a passive boost to the adaptive immune system. So the adaptive immune system is your backup system, and it attacks invaders if they get in. They should be blocked at the surface. They should be recycled in the innate immune system. But for most of us, our adaptive immune system, our antibodies and T cell responses, are very active, called upon regularly, and they are very energy requiring. So we need to nurture them, and we need to understand their nature. Now, a healthy immune system has no allergies. There is tolerance. It's called homeostasis or self-regulation. you neutralize infections and allergens, you repair from daily wear and tear, and there are 50 billion of cells, dendritic cells, white blood cells, that each are capable of recycling 50 foreign invaders. In fact, there's an old-fashioned test called phagocytic index that you might want to do to confirm to people that their innate immune system is weak because it lacks essential things that it needs, and it's got too much of the toxic anti-nutrients. It's very energetic. And so there is a clinical test called phagocytic index if you want to measure the function of the innate white blood cells. And you will often find that they are present, you can count them, but they're not very functional because they lack enough of the good stuff. They have too much of the bad stuff. They lack essential nutrients. They have too much of the antitoxin, pro-oxidative, harmful molecules. And we end up then with an overburdened immune system that is doing too much defense work, usually with a lack of deep restorative sleep, and therefore a lack of repair. So there's a critical need to tune up most people's immune defense and repair system, most people's immune system, now. Now, the 21st century is more challenging to every aspect of biology than in any recorded time we know. This is a back to the future approach. It's based on classic wisdom as well as current evidence, the functional, the integrative, the holistic, the comprehensive, the really causal, looking at the causes, not at the consequences kind of science. And part of the reason that this is so important is because hidden immune burdens, the delayed reactions to foods, chemicals, medications, and environmental substances, it takes from 3 hours to 3 weeks after exposure before you get symptoms. Look up Arthus reactions, Schwartzman reactions, serum sickness reactions. We have a whole literature that most of us as clinicians are not too familiar with, but about the long delay in the adaptive immune system between exposure and consequence. Now, these reactors drain vitality. The patient comes in or the person comes in and says, my get up and go got up and went. I can't find it, and I'm slipping down the slippery slope of fatigability, irritability, lack of concentration, lack of productivity, and help me. And we come in peace to help. And we start with looking at hypersensitivity, looking at delayed allergies. And we know about the immediate reactions. These are type 1 IgE reactions. They are immediate, which means if you get stung by the bee and you get hives or wheezes or itching or anaphylactic shock, it was probably the Hymenoptera venom to which you are immunologically sensitive. Then we have the delayed. And now we have 3 different pathways. We have type 2 reactions, which are antibody, could be IgM, IgG, or IgA. Then there's immune complexes, and then there are T cell responses. And you can measure all of that in a mixed cell culture. And people have been doing lymphocyte cell culture for many years. People had been doing ELISA amplified procedures for many years. What we did for the first time in the early 1980s was bring those 2 technologies together, have an ex vivo lymphocyte response assay in one step with an amplified reaction and with good reproducibility. That is, and we'll talk about this later, less than 3% variance on consecutive blind split samples. So we have to look at all delayed pathways concurrently. They are hard to identify by history or even by serology, that is serum antibodies like IgG tests. You can see if an IgG antibody is present. You can see if it is specific for a substance in question. But you don't know if it's helpful or harmful. You don't know if it's complement fixing or neutralizing. You don't know if it's a good or a bad antibody. A lymphocyte can tell you, because lymphocytes react to bad antibodies, that is complement fixing reactive antibodies, but not to neutralizing protective antibodies. So another reason to do an LRA, a lymphocyte response assay, And we had the privilege of developing the first one-step, high-precision, reproducible, ready for prime time, and shippable, because the specimen is taken in ways that do not activate or damage the specimen. We then keep it cold, but not frozen, basically 4 to 10 degrees centigrade, so it's cold and very little metabolism takes place, but not so cold that ice damages the cells. And then when we get it to the laboratory, we gently spin out the red cells. We get the cell-rich plasma. We bring that back up to body temperature for about 3 hours. And then we look at how the antigen-presenting cell has given the processed antigen to the white blood cell. And that white blood cell then lights up. It goes into mitosis. If you waited 3 weeks, you'd see a colony of cells. If you used radioisotopes like radioactive thymidine, you would see radioactive incorporation within 2 or 3 days. But we look even further upstream than that at the MHC locus, a specific kinase, a phosphorylating enzyme that sends the nucleus a message to start dividing the cell, but it also changes the whole charge distribution on the surface of the cell, and that's what we see in the laboratory. If you look on the left of this next slide, you will see non-reactive lymphocytes. Some people say that looks kind of like Starry Night as a painting. And on the right, you see the reactive lymphocytes that have, quote, lit up. You see a rim of opacity. You see a halo around them, and it's symmetric. And our technicians spend months and months learning how to read this. We compare them with each other to make sure that everyone reads and gets the same results. And that's what we've been doing for over 80,000 cases over now 3.5 decades, and still continuing to add to the science and to the clinical understandings. Now, just to review very briefly, on the left of the slide, you see the standard ELISA 5-step, say the IgG ELISA. It's a 5-step sandwich. This is the Burston and Yalow methodology from the 1950s. There is 3 to 4% variance added for each step, which means the best you can do from a conventional IgG test is 15 to 20% variance, which is fine for research and epidemiology, but is not precise enough for individual case management. Now, on the right, you see our approach, the single-step LRA that brings together enzyme amplification, the kinase, and ACT cell culture in a novel microtiter well and plate that accomplishes, quote, the impossible. It is thin like a slide that you can look at in a microscope, but it's deep enough to allow us to incubate the cells where the antigen or the test has been coded on the bottom, and then have enough cells to look over the well surface to get an accurate measure of how many of the cells are reactive. Now, I mentioned several times precision, and now I'm going to burden you with a little bit of an abstract that we presented at the American Society for Investigative Pathology, Experimental Biology a few years ago. And this is consecutive blind split samples over 3 years, no selection of data, consecutive blind split samples, and matched were 97.6 and unmatched were 2.25%. And what we say is less than 3% variance on blind split samples, which means we're reproducible. Then we have to put this to a test in an outcome community-based study to see if the results are meaningful, and we have done that. But we first want you to know that they're accurate, because many immunology procedures have such a wide variance that when you do blind splits or when you split the results on the same specimen, you get significantly variable, different results. Now, in regard to our LRA, because it's so miniaturized, because on just microliters of each specimen we can see a result, we can, on 1 ounce, about 30 cc's of specimen, look at over 500 cell cultures, if you ask us to, for foods, additives, and preservatives, and we have the largest number of those. We actually standardize all of our own antigens. We purify them and standardize them because I'm a biochemist. We have the most environmental chemicals to test, 60. We have toxic minerals known as heavy metals, 14. We have medications, mold, danders, hair, and feathers, and herbs, because all of these could, any or all of these could be sources of immune burden for an individual. So this is a personalized procedure. It has good science and evidence. It has good outcome studies to support it, and it is ready to be first line for comprehensive care in the 21st century. And what do we recommend? You start with the high sensitivity LRA. The immune enhancement program involves substituting as carefully as possible, including in hidden places where you might get exposed to that substance, for 6 months for strong reactions, at least 3 months for moderate reactions. During that time, you correct nutritional deficiencies, reduce defense and repair, abilities. If you're deficient, your immune system and hormones and neurochemicals aren't as effective. They aren't even produced in adequate amounts. So we work to correct nutritional deficiencies over those months. We initiate a detoxification enhancement mechanism inside the person. We alkalinize, it's called the alkaline way for a reason, to enhance human healing responses. It turns out that the body produces extra acid every day. That's a fact. And we must take in buffering minerals, particularly magnesium, which is hard to get in order to neutralize those acids. Otherwise, we slip into metabolic acidosis and chronic disease. Now, we've also personalized our approach. And so we standardized many years ago an HAQ, a health appraisal questionnaire. It's 2 sides of 1 page. People score themselves on a variety of questions. And this then allows us, with our smart system, with our AI smart system, to recommend essential priority supplements and elective supplements, to include activity and environmental recommendations to improve their health quotient, and a rotation diet option is available. And it all comes down to alkalinization. A colleague of mine wrote a book called Alkalinize or Die. That's not far from the truth. And so we produce a personalized report, digital. We can do it physically. And it includes the results, including the why and the wherefore, and in regard to the items that are reactive, how to substitute, because this is a window of opportunity to evoke healing responses. It's not deprivation. How to substitute, and more importantly, where you might be exposed to this unexpectedly. Now, we felt the obligation, and years ago we did outcome studies, starting with fibromyalgia pain, and moving on to type 2 diabetes and type 1 diabetes. Fibromyalgia as a mystery condition of unknown, quote, cause, and diabetes being the classic autoimmune condition. And basically the goal was to restore tolerance and reset hypersensitivity based on this personalized testing. And when we went out, a third party had to confirm the diagnosis, and the only criteria for participation was willingness and ability to comply. If the person could say, yes, I will do my best, they could be included in the study. And we had a question and answer time every 2 weeks. We were available to answer questions either by the clinician or by the participant. And if we look at the results, a novel treatment for fibromyalgia improves clinical outcomes in a community-based study published in the Journal of Musculoskeletal Pain by Professor John Russell. And we showed in just 6 months, and every one of these people had been multiple failures. That was part of the screening criteria. They had expectations that this wouldn't work. And yet there was 50% less pain, 70% less depression, 50% more energy, 30% less stiffness, as rated by both the participant and their independent clinician. And we published this many years ago, but are happy to share that with you, and the follow-up that says these people continue to feel and function well and spend less. If we look now a bit at the data from that study, we normalized the before, we normalized the baseline to 100%. So all three groups were at 100% before. At the midpoint, all three seem to be getting better, but at 6 months, you see the placebo group on the left, that's kind of the magenta column, that's slipping back almost to baseline. The other two are getting better. Primary fibromyalgia means you only have unexplained muscle pain. Secondary fibromyalgia means you have other chronic ills with it. And because the secondary fibromyalgia people had multiple things corrected, they proportionately did even better than the people with primary fibromyalgia. What about diabetes? The article is titled Tolerance Loss in Diabetics, Link with Foreign Antigen Exposure, and the result, the bottom line, the takeaway, is a more than 1% decrease in hemoglobin A1C levels in both type 2 and type 1 diabetics, in diabetes, and an 18% reduction in insulin levels in type 2 diabetics, which means their insulin function improved, their insulin resistance reduced, their insulin tolerance got better. When we look at the data from those two diabetes studies, and the type 1 is in a kind of sandy color, and the type 2 light blue, we see there were similar proportions of reactions to sweeteners. And by the way, you're sweet enough as you are, you don't need to add empty calorie sugar or artificial sweeteners to your diet. Toxic minerals were very present. The reactivity to toxic metals in type 1 diabetics was over 50%, but only 15% in the type 2. Environmental chemicals were richly represented in both. Additives, which are very important in terms of immunology and hormones and chemistry. 80% of the type 1 and 46% of the type 2 reacted to additives. But interestingly, cow dairy incidence of reactivity was much more common in type 2 diabetics than in type 1. And part of the issue in relation to digestion and metabolism, the microbiome and the metabolome, is leaky gut. In the gut, there are things called Peyer's patches. There was a professor named Peyer, and he described the irregular large surface area given the tube nature of the intestine. And when I was a young physician scientist, I was taught that the Peyer's passage were vestigial or vestigial, if you were more academic. And in fact, they're very important in digestive function. And on the left, you see a healthy intestinal lining. On the top right, you see a damaged intestinal lining. And the consequences on the bottom, the leaky gut, which means there are little spaces between cells that foreign stuff and partially digested reactive stuff can wiggle through. The cells themselves are not electively producing protective molecules like secretory IgA and mucins and opsins and defensins, because we're not giving them enough of the good stuff and we're giving them too much of the bad stuff. And these lining cells of the intestine, known as enterocytes, because they line the interior, the enterocytes, are where our digestion happens. And if we nurture it, it will come back and restore. But the typical American has significant and profound maldigestion, dysbiosis, and other similar maladies. Now, what are the microbiome essentials? Dr. Dennis Burkett taught me prebiotic fiber, that is things you have to chew or take as supplements of prebiotic fiber, 40 to 100 grams a day, probiotic bugs, 40 to 100 billion live bugs a day from fermented foods and supplements with active CFU, colony forming units, active bugs. And the third essential category are symbiotics, recycled glutamine. You recycle it with PAC. You take 1.5 grams on rising and before bed, and maybe half an hour before exercise, to evoke healing responses, to energize the enterocytes who use glutamine. But you don't want glutamine to go to glutamate. So you recycle it with PAC, you get the benefit of the glutamine without ever building up glutamate. On the next slide, we have a visual representation of the digestive microbiome. Food goes to energy. Eat what you can assimilate, utilize, and eliminate without immune burden. And we recommend, as one of the 4 self-assessments, the GI transit time. You use activated charcoal. And when you swallow those capsules, you mark the time. And when the black crumbly stuff comes out of your tush, you mark the time. And the difference in hours is your digestive transit time, and a healthy digestive transit time is 12 to 18 hours. Well, now let's turn to some clinical examples and outcome cases. Ulcerative colitis, an autoimmune condition, a chronic disease. In this case, a 31-year-old woman. She had done an over-the-counter cleanse that triggered bloody stools. We don't recommend that. She did have a positive family history, and she was treated with Lialda without success. And then we move on to our therapeutic plan, which included the LRA tests, and there were 18 reactions. The plan was to substitute as best as she could after counseling, include supplements based on the HAQ Health Appraisal Questionnaire. And in her case, that included a super multivitamin, multimineral, essential omega-3, 6, 9 fats, bone-building nutrients, recycled glutamine with PAC, that's the symbiotic, comprehensive probiotic blood-building complex with ferrous aspartate, the gentle iron healthy source because of her iron deficiency, hydroxycobalamin and polyphenolics. And 1.5 years later, the outcome was a normal CBC, still on Asacol, but reducing slowly. Weight has come back up. Still amenorrheic, but breast tissue's coming back. Gets acupuncture monthly. The family is healthier. The husband lost weight. The kid's eating healthier. And they're going to repeat the 6-month cycle soon. Now we look at adrenal immune connections, and we see on the left side a cycle. If you start at the top, you have the paraventricular nucleus of the hypothalamus that feeds, that sends signals to the anterior pituitary. Now today, we really know it's the pineal that sends the message to the paraventricular nucleus to influence the pituitary. The anterior pituitary provides the ACTH for the adrenal glands that produce the glucocorticoids and other factors that influence the immune system, and there is feedback. So we're careful to give the essential nutrients, but not the products that feed back and cause inhibition. So we work with both physiology and biochemistry. We work with clinical experience and with evidence. And as examples of where you might consider this issue, people with chronic stress, and that's 80% of why people seek out medical help. They have low circulating cortisol and or a dysfunctional HPA axis when you look at saliva at 4 times in the day for cortisol and DHEA. It affects immune system adversely. It diminishes cytokine regulation, increases the chance of a storm in cytokines. It increases inflammation repair deficit. There's a dissociation between plasma concentration of ACTH and the glucocorticoids because of the distress and the exhaustion and the overstimulation that's going on. And it has to do with glucocorticoids and catecholamines and their regulation. And you see on the bottom right, cytokine expression, immune cell activation, bacterial proliferation. If you want all those to be in harmony and communion, then our approach is the most evidence-based around. A different case. This is now ADHD and energy depletion in a 50-year-old woman. Lack of restorative sleep, decreased energy, weight is up, and mild alopecia. She had taken Adderall, although without much benefit. The LRA tests found gluten, titanium dioxide, sulfite, and salicylates. She was asked to substitute for those few but important reactors while taking a personal supplement regimen and an alkalizing lifestyle. After a month, she achieved appetite control. After 2 months, sleep rhythms returned to normal and she was getting restorative sleep. After 3 months, hair loss had diminished, night tremors were gone, prescriptions were down by 50%. And after 4 months, energy level high, sleeping well, focus improved, and she has continued to improve. On the next slide, we look at the overloaded immune system. At the top, we see inflammation, which is really repair deficit. And in this case, we're talking about the thyroid gland, since it is so often the focus of repair deficit. And as you see on the bottom, when you don't repair the blood-thyroid barrier, when you don't provide all of the essentials that the thyroid and the blood-thyroid barrier need, you end up with a repair deficit and what's called autoimmune, self-attacking thyroiditis. And now we have a case of Hashimoto's thyroiditis in a 30-year-old woman. Took 7 years for her to suffer before she was properly diagnosed. She had fatigue and muscle weakness, joint pain and weight gain, bloating and digestive distress, mood disorders, and so forth. She was treated with Synthroid, T4, with modest benefit, Euthyrox, which is a European version, and then got to our clinician, our colleague, who did the LRA tests, identified 20 out of 375 tests that were reactive, 20 substances to substitute for in her case. And it was the same approach. Substitute with guidance, targeted supplementation, diet, hydration, attitude, activity. And the initial outcome was pain reduced and thyroid medicines reduced, and quote, the best that the person has felt in years. And the colleague who managed this case said, it's that kind of comment that they hear so often when you follow this approach, the alkaline way of evidence-based care in the 21st century. It's that kind of comment. This is the best I have felt in years. Warms the heart and makes people who provide that care know that they are making a difference. Second case of thyroiditis, 36-year-old woman, 5 pregnancies and 3 children, low energy, weight is up 18 kilograms, little joy in life, morning temperature's low. And Broda Barnes pointed out that if you accurately measure temperature before you get out of bed, you can often see a low temperature that correlates with thyroiditis at different stages of the thyroiditis continuum. They measured total T3 and T4 and they were unremarkable, but when they looked at free T3, it was low. Free T4 was within lab range. TSH was up, not surprising. Healthy TSH is between 0.5 and 2.5. The labs often have a wider range for statistical but not clinical purposes. There was an increase in 2-hour postprandial glucose and insulin, an increase in free cortisol, and a decrease in DHEA, which says a dysfunction in the hypothalamic pituitary stress hormone response. And the person substituted for their reactive substances following an alkalized lifestyle that did include briefly 2 grains of desiccated thyroid if the morning temperature was below 98 degrees Fahrenheit, the use of green dichromatic lights, which we often recommend and can teach, salt and soda baths as part of preparing for restorative sleep. And we can also explain any of these particulars to any colleague that wants to go into more depth. 6 months go by of best effort on the patient subject's part, asymptomatic by report, lost 10 kilograms of weight, 22 pounds weight has reduced, discontinued thyroid hormones, free cortisol and DHEA, glucose and insulin have all now normalized. So this is using physiology and restorative care before pharmacology and symptom reactive care. Now, just a footnote, the 21st century is different even than the 20th or the 19th century, and there has been at least a 15-fold increase in industrial chemicals. All pregnant women, when their cord blood is tested, have at least on average, at least 43 xenotoxins at low levels, but we don't know the interactive effects or the long-term effects of these, what we would call forever molecules, these anti-nutrient, oxidative, pro-oxidative, antioxidant depleting chemicals. So what's a person to do? We start bio-detox with superfoods. This includes garlic, ginger, onions, broccoli, sprouts, and eggs, GGOBE. If you follow this, you will get lots of sulforaphane and IP6 and other beneficial things. And we recommend that you take a look at our writings or have us send you links to YouTube channels and to videos about the proper use of garlic and ginger and onions and brassica sprouts and healthy eggs as staples in the diet. President Thomas Jefferson suggested that we make these staples out of condiments and condiments out of staples if we wanted to lead a healthy, longer life. And then the glutamine recycled by PAC, as we talked about before, for digestive energy and restoration. And now we get to the question of how much ascorbate vitamin C does a person need based on their oxidative stress or burden. And we many years developed and recommend that you include what we call the ascorbate calibration, or proverbially the C cleanse, capital C cleanse. And if you're healthy, you start with a half a teaspoon. If you're moderately ill, you start with a teaspoon. If you're chronically ill, you start with 2 teaspoons every 15 minutes in an appropriate amount of water or herbal beverage. And within a finite period of time, for most people within a few hours, you will have what we call a cleanse. And most people want a cleanse. You could also call it a flush, and we did originally call it the C flush. It's the same thing, but more people will do the C cleanse than the C flush, even though it's just 2 different words for the same technique. So we pioneered this issue of letting individuals find out on a weekly basis how much ascorbate they need, of course, using only the fully buffered, fully reduced Nature's L ascorbate, which means it was produced under a nitrogen blanket, something that we helped pioneer to protect the delicate antioxidant in production. So we recommend the C cleanse as one of the self-care essentials. And if you look together, here are reports that we received that we've summarized in some of the chapters that we've had in the Watson and Preedy series on bioactive foods and dietary interventions, this one in cardiovascular disease. And here you see the self-reports of how much ascorbate it took to cleanse. In yellow, the people who are asymptomatic; in orange, the minimally symptomatic people; in green, the typical ambulatory walking wounded; and in red, the unwell, significantly unwell. So sometimes it takes a lot. Our team can help coach you in how to do, what to do, and when to do. On the next page, we look at the alkalinizing foods. We look at food effects on body chemistry. This is a chart we made up many years ago and have continued to improve upon. You want 80% of what's on your plate to be alkaline forming, in this case on the right half of this chart, and 20% or less during recovery on the left side. And the result is that you can modulate or reduce the adverse effects of the acids of metabolism by taking in the alkaline minerals in forms that can get into the body. And we, of course, have pioneered the foods that have better bioavailability, especially organic and biodynamically sourced, but also supplements that are all active, full disclosure, no hidden components or allergens, and most importantly, been used in successful community-based outcome studies. So alkalization brings healthy balance from foods and water by taking in mostly alkaline forms. You should be active and check your first morning urine pH and adjust your magnesium choline citrate intake based on that. You want to do abdominal breathing like a baby for 5 minutes twice a day to improve your diaphragmatic stretch on your intestines and lungs. You might want to use green light, either by forest bathing, that is ambling under a green canopy in a mature woods, or a green dichromatic light. You want to eat in harmony with your nature and lifestyle. You want to eat locally grown, vine ripened, organic, or biodynamically derived foods as possible. You want to make restorative sleep a priority, and we have a half-hour pre-sleep program to help you detox, sleep better, and get some active meditation and abdominal breathing all at the same time. And you want to work your muscles and relax in proportion. Very often people over-exercise and they don't stretch enough. They don't balance their activities or their attitudes. They don't balance their diet and their hydration, and we help them restore balance and reap the benefits. Now, that first morning urine pH is a predictive biomarker, one of the 8. It gives us the status of cell acids and minerals, the enzyme catalysts that are very exquisitely pH or acid-alkaline sensitive. So when you're alkaline and energetic, your protein efficiency is more than 90%. And when you have metabolic acidosis, it's less than 10%, which means a lot has to get recycled, and that's a terrible burden on the cell metabolism. Now, magnesium is the counterbalance to calcium. It's known as the forgotten electrolyte. We want you to remember it because it's so important for bones and joints, for blood vessels and brains, for cell communion and communication. Now, that urine pH after rest, if you're too acidic, if you have metabolic acidosis, it'll be below 6.5. And you can go up and go to the bathroom, but you can't go to the gym or the kitchen. You need 6 hours of rest for the fluid to equilibrate in the bladder, and you want to be in the healthy green 6.5 to 7.5 range. And you want to increase your magnesium choline citrate if you're below 6.5, 1 dose for every half pH unit. And if you're too alkaline, you might be in a catabolic state and tearing down your lean muscle and bones, and we want you to reverse that with our guidance and help. Now, in terms of biomarker solutions, we mentioned magnesium, but it's got to be bioavailable, and you have to enhance the uptake with choline citrate. No other choline works, no other salt works, so choline bitartrate doesn't work, choline citrate does work. And we recommend 440 to 880 milligrams of elemental magnesium daily because of all the wonderful things that magnesium does. And then you get the benefit from choline of a soothing neurotransmitter called acetylcholine and more soluble, more liquid bile coming out of the liver and gallbladder. The citrate energizes on alkalinizing and alkalinizes. It's all active and all beneficial. So magnesium is nature's stress buster. I'm just reminding you of that. It protects the brain from depression and other adversities. It reduces repair deficit, does lots of good things. I mentioned about restorative sleep and how important it is, and it is a choice. Here you see, and you may want to come back and review the salt and soda bath, the abdominal breathing, the dichromatic green lights, the no electronics in the bedroom policy, the no caffeine or heavy metal exposure in the bedroom. You might want to use the enhanced uptake tryptophan that we have made available so that the tryptophan goes into both the gut and the central nervous system and produces serotonin and melatonin as needed. Now, I mentioned the 8 predictive biomarkers. I want to briefly review them. We have whole webinars on the details of this, but hemoglobin A1C should be less than 0.5. HSCRP should be less than 0.5. Plasma homocysteine should be less than 6. The HSLRA, the lymphocyte response assay, should be tolerant, have no reactions. Your first morning urine pH should be between 6.5 and 7.5. Your vitamin D should be between 50 and 80. Your omega-3 index should be greater than 8%, and your 8-oxoguanine should be less than 5. And these 8 predictive biomarkers cover all of epigenetics. and they are the logical suite to do after the 4 personal self-assessments. So in regard to viral infection, first-line defense, if your immune defense and repair system is working electively and protectively, your mucosa surface protects you, the innate immune system protects you, the adaptive immune system is in reserve. So whether we talk about any kind of a virus, the viruses like you to be low in antioxidants and have a high oxidation reduction potential. We want you to have enough ascorbate and other antioxidants to have a low redox potential and turn off the pathogens of viruses, including the prions and the parasites. If you feed them metabolically, they will proliferate. If you stop feeding them, they will go away. That's physiology before pharmacology. That's nature, nurture, and wholeness as we apply it. Now, if you need the adaptive immune response to a viral infection, it is mobilized when called upon. It produces antibodies through lymphocytes called B cells. It's called a humoral or cell-mediated response when that happens. T cells react as part of that without antibody signaling. And they do distinguish what is foreign from what is not. Foreign from self, you from not you. Very important for you to be you and things that are not you to be recognized by your immune defense and repair neurohormonal systems. So we want to reduce risks, boost immune defense and repair, and stay healthier. Hydration, first morning urine pH, restorative sleep program, the digestive solution through the LRA plan, air quality, including air cleaners if needed, and always include nature's buffered reduced L-ascorbate, magnesium and choline citrate based on acidity or metabolic acidosis signs. Most people need zinc one way or another through zinc-rich foods and or supplements that are bioavailable. Polyphenolics are getting more and more important, quercetin dihydrate and low molecular weight. LMW is low molecular weight, OPC, orthoproanthocyanidin. That combination we pioneered in the late 80s as a safer, more effective, more bioavailable flavonoids and flavanols, the polyphenolics. We have done chapters in textbooks on the subject. Happy to send them to you if you would request. And we do include liver and detoxification support in most cases. So this immune system tune-up has to do with an amplified response to foreign invaders, and more foreign invaders than there used to be, and they tend to be more long-lived. That is, the chemicals that are anti-nutrients. Some of them are forever molecules that are hormone disruptive. We rethought inflammation as repair deficit. We know how to reduce the toxic burden and increase the essential nutrient intake. We want to pay attention to delayed allergies through the ex vivo HSLRA test. And we want to have a comprehensive program of detoxification and immune restoration based on what people eat and drink, think, and do. So it is about personalized lifestyle and proactive primary prevention and practice. So thanks for your interest. And hopefully you will find Dr. Russell Jaffe on YouTube. You will find the PIH Academy and our other continuing education programs worthy. And that we will together help speed the transition from the current sick care symptom reactive system to today and tomorrow's cause-based, evidence-based, personalized medicine that is proactive and truly preventive. So thank you. May we all be well and happy.